Now we're going to talk about the control of the respiratory system. And this is something a lot of exercise physiologists are interested in. And their focus is on how physiological systems respond to changes in activity. And that could just be walking around, or it could be very intense exercise performed by professional athletes. So this experiment that we see a screenshot from on the left is an example of a case where you have a subject that you can ask to ride. He's on a bike. You can ask him to ride at different levels of exercise intensity. And that's going to manipulate the oxygen that he consumes because aerobic metabolism is dependent on oxygen consumption. So as we move along towards the right of this x-axis, which is achieved by increasing exercise intensity, then that's going to drive an increase in oxygen consumption. If you instrument your athlete so that you can monitor the dissolved gases in the major veins and the major arteries, then what you, not surprisingly, would see is that the partial pressure for oxygen drops at these higher exercise intensities. Now, what's impressive is that the control system for respiration, and circulation for that matter, is so effective that on the arterial side, there is virtually no change in the partial pressure for O2. It's maintained around a set point of 100 millimeters of mercury, despite the fact that there is less and less oxygen in the blood as it returns to the lungs at the higher exercise intensities. So at close to 100%, we have significantly less partial pressure in the blood that returns to the lungs, yet the lungs compensate and increase their uptake of oxygen in order to maintain that partial pressure around 100 millimeters of mercury. So we can say that this respiratory control system does that. It maintains the partial pressure for O2 against the increased aerobic demands of higher exercise intensity. And so the focus of this lecture is on addressing how. What are the components of the control system that allow for this impressive feat? Now, if we perform an experiment like the one we see on the left, then we can monitor, first of all, what the respiratory system is doing in response to exercise intensity in order to help achieve that constant partial pressure. What we see there is something that's impressive, especially at the really high intensity exercise levels, because the ventilation rate has a nonlinear relationship with respect to oxygen consumption. So at high intensity, it is really cranking up the amount of ventilation in order to keep that arterial partial pressure of O2 constant. Now, for the control system, we've already talked about a lot of the pieces on the efferent side of things. That includes the skeletal muscles that drive ventilation. So we have, for inspiration, where the goal is to increase the volume of the thoracic cavity, the diaphragm muscle that provides a floor for the thoracic cavity contracts, and we have both the external intercostal and the scalene muscles expand the volume of the rib cage, or the volume within the rib cage. And when they become activated, you have that drop in pressure in the lungs, which drives the flow of air into the mouth for inspiration. On expiration, that all reverses. We have a relaxation of the diaphragm muscle and the contraction at high intensities of the abdominal muscles, and at lower intensities, the internal intercostals suffice to drive expiration. So that's a reduction in the volume of the thoracic cavity, which in turn elevates the pressure on the inside of the lungs. If that pressure is high, then the air outside is lower. That means air flows through the respiratory tract, and we have an exhaling of a tidal volume outside the mouth. All right, so we have two sets of muscles, one that drives inspiration, the other one for expiration. These are two phases of ventilation. Now, what about the sensory side of things? Much like the sensing that was involved in the control of the circulatory system, the respiratory system has sensory organs on the arterial side that monitor blood chemistry. And they're located in the same positions. So we have in the carotid artery a group of chemoreceptors that monitor the dissolved gases. And in the aorta, we also have a clustering of those chemosensory cells as well. So in the carotid artery, we have something called the carotid body, and then around the aorta, we have the aortic body. In both cases, glomus cells are included in those bodies, and that provides the means for measuring dissolved gases in the blood. And so what we have here is for a capillary, but this could be a larger blood vessel. We have a glomus cell that's in close proximity. And this glomus cell is sensitive to the chemistry or the dissolved gases in the blood. And in particular, we're going to illustrate the sensitivity that this glomus cell has to oxygen. And the way that this works is oxygen freely diffuses across the cell membrane of the glomus cell. And if the partial pressure is low, then that influences the permeability of the membrane to potassium. So if we reduce potassium permeability, that will depolarize the membrane. That in turn triggers a calcium voltage-gated channel, which in turn activates the release of a neurotransmitter. Okay, so this is similar to what we see on a presynaptic axon at a chemical synapse. So calcium influx triggered by this depolarization driven by potassium causes a release of the neurotransmitter, and then that is in turn excites an afferent postsynaptic neuron. All right, so what this illustrates is the ability to monitor oxygen on the arterial side. So oxygen is an important sensory cue that's picked up by these chemoreceptors that are in the periphery, these peripheral chemoreceptors. Now, the respiratory system is also informed by sensing in the brain itself. Those are central chemoreceptors that are found in the medulla oblongata. And that operates by a slightly different mechanism. So there we do have capillaries. All of the neurons in the brain are separated by, or you could say that they're bathed in a cerebral spinal fluid. So the chemistry of the cerebral spinal fluid can act directly on the neuronal cells in the brain, and therefore these brain cells are not directly sensing what's in the capillary. So let me illustrate. These chemoreceptors are sensitive to carbon dioxide. So the carbon dioxide in the capillary will diffuse into the cerebral spinal fluid, and then that causes the chemical reaction that we've described in an earlier lecture when I was talking about carbon dioxide transport. That is, carbon dioxide can be converted into first carbonic acid and then ultimately bicarbonate and protons. So that's going to lower the pH. The protons produced by this reaction are then detected by receptors in the medulla. And that's going to stimulate the integrating center, which I'll describe in a moment. Okay, so we have both in addition to the peripheral chemoreceptors, these central chemoreceptors located in the medulla oblongata. So we're going to add that level of control. It allows for sensing of carbon dioxide. Now, pH, which is influenced, as we see in this reaction here, by the partial pressure for CO2 is also influential on, or I should say, it affects the glomus cells as well, although not as strongly as oxygen. All right, so we're going to add pH as sort of a secondary. I'm sort of making it not as bold here because it's sort of a secondary influencer, but yeah, pH changes are detected by the glomus cells as well. Okay, so that's the afferent side of things. Both the peripheral and central chemoreceptors, they in turn inform the integration centers, and there's multiple integration centers here for the control of respiration. Now, this is a little more complicated than the integration centers that we've considered so far. Okay, they are located in the brain stem. All right, no surprises there. We've already mentioned the medulla oblongata. Not only does it serve a sensory function, but it also plays a role on the control of the efferent side of things. And in particular, there are two regions that matter for the respiratory system. There's one that's more dorsal. Okay, so on the drawing, dorsal would be towards the right. And the dorsal ventilatory group, just think of a cluster of interneurons, are responsible for activating the skeletal muscles that drive inspiration. So you have descending commands, ultimately a chemical synapse to motor neurons that activate those muscles. So DRG is the dorsal ventilatory group, which is activating those muscles. So not surprisingly, we have something similar, another clustering of interneurons in the medulla oblongata that activates the muscles responsible for driving expiration. Those are known as the ventral ventilatory group because they're relatively ventral in the medulla. All right, well, this is puzzling. You've got two sets of muscles activated by two different integrating centers. These integrating centers need to coordinate. If you were to activate the abdominals the same time as the diaphragm, then you would have problems because you would essentially be driving the respiratory system, driving the changes in the volume of the lungs in two different directions simultaneously. So instead, the interneurons between the dorsal and ventral respiratory groups need to facilitate a coordination so that the abdominals and the internal intercostals that drive expiration are not activated at the same time as all those muscles that drive inspiration. All right, they have to be out of phase with each other. And the way that this is achieved is through a network of neurons between these two groups. All right, that network of neurons is known as a central pattern generator, a CPG. Okay, so the dorsal and ventral respiratory groups function as a central pattern generator. That is, there are neurons that make it so that when the ventral respiratory group is activating these muscles, then the dorsal respiratory group is inhibited. And when the dorsal group is activated, then the ventral group is inhibited. So we can say that generally CPGs are networks of neurons or interneurons that produce spontaneous oscillatory patterns. Okay, they're sort of like the autorhythmic cells, but autorhythmic cells, just a single cell can produce an oscillatory pattern. Here we have a network. We have a group of interneurons that achieve this function. And it's critical, again, for the reason that we do not want to activate the inspiration muscles at the same time as the expiration muscles. Now, in order to vary the rate of respiration, then we're going to introduce a third integration center. And it's also in the brainstem, but in a different region. Okay, this region known as the pons. The pons varies ventilation in response to input from the chemoreceptors. Okay, so it's the pons that's the receiver of the peripheral and central inputs. But then its job is to modulate the CPG so that the DRG and the VRG are not activated simultaneously. So we're going to add our third. This is the final integration center, and it's receiving the peripheral and central chemoreceptor inputs. And it's comparing those against set points in its decision making about how to vary the CPGs. Okay, so it's that integration center that operates according to our algorithm. And for any integrating center, it's implied that it operates according to an algorithm. So we're going to focus on the pons here. And for the pons, we have one algorithm for the case of hypoventilation. All right, so remember, this equation means we have a particular state variable compared against a set point. And when the difference between them is below a threshold, then we're going to execute some kind of action. All right. And in this case, it's stimulated by an elevation in the partial pressure for CO2 and a depression in the partial pressure for O2. All right, now let's think here. Hypoventilation this means that we're not ventilating enough to keep up with metabolism. So it makes sense that our set points, and we've got two set points here. One of them is the partial pressure of CO2, or you could say that there's two state variables, and they're simultaneously acting. So we have the partial pressure for carbon dioxide, the partial pressure for oxygen. Carbon dioxide is detected both by peripheral chemoreceptors. That mechanism we did not detail, but it's similar in its action to how the glomus cells respond to oxygen. So carbon dioxide acts in the periphery. It also acts centrally. So in the medulla oblongata itself, it detects those changes in carbon dioxide. Now, in hypoventilation, we have too much carbon dioxide and not enough oxygen. So that drop in the oxygen does activate the glomus cells in those peripheral chemoreceptors. So that's the state variable in the case of oxygen. That is then compared against a set point. So we have set points for both oxygen and carbon dioxide in the periphery. And the pons makes a decision about how to respond to that. And in particular, it's going to increase ventilation. It's essentially going to drive the CPG to greater frequencies of activation. So that's the algorithm for hypoventilation. So I sort of mapped out the path for oxygen through the periphery, but we have the same thing going on for carbon dioxide in the central chemoreceptors. There's a partial pressure for CO2 set point, the error in the current state of carbon dioxide compared to that set point, and then the pons makes a decision about increasing ventilation. Hyperventilation is the opposite problem. That is, we're ventilating more than metabolism requires, and so our error is greater than some threshold. It's stimulated by the partial pressure for CO2 being very low, the partial pressure for oxygen being very high, similar kinds of pathways. The pons, in response, decreases ventilation through the CPG. And that's the control system. So that is the answer to our initial question of how the respiratory system manages to keep the partial pressure for O2 constant against that increasing demand of more intense exercise that drives an elevation in metabolic rate.